Um, so thanks for having me here, and I'm going to piggyback a lot on what has been discussed this morning and synthesize what Joanna was showing about the modes and waves and models. So I'll try my very best, but if it gets boring, I'm sorry. How do I queen? Perfect. Okay. So because it's an educational session, I'm not going to focus a lot on results and stuff, but I'm going to gloss over them. But mainly, I really want to cover a lot of the kind of the fundamentals and the basics on what the modes are and why they're important, what do they represent. I'm going to talk a little bit about neural field theory, which Michael Breakspear and Vito Jessa this morning kind of introduced, but I'm going to discuss it a little bit more in detail, and how it can be related to, and what are the properties of these modes, and so on and so forth. And I'm going to show a little bit of how we use them in one of the publications that we had last year, and what are the exciting applications that we have. These are just some of the things that I'm going to show. So hopefully it will be a worthwhile experience for everyone. And if you have questions, yeah, talk to me later. Right, so let's start with I always like starting with this one, because again, as Joanna was saying, it's rare that we look at functions. This is functional data, fMRI data in both space and time. We always reduce it to parcels and just look at time series of all those parcels. But we never look at actually what the data looks like in both space and time. So if you actually look at, let me go back just to play the video again. Just to show you, I didn't do anything on this. This is just HCP data put on the surface and not do anything. So just visualization. If you actually look at the data, you can really see on a meso and macroscopic scale, which is what fMRI is trying to capture, you can really see this almost continuous like propagation of dynamics, which obviously, as Joanna said, you lose all that information if you partition the brain into discrete regions and look at the time series. So it's really important that we do not forget that, and we also try our very best to explain these types of continuous dynamics. And these continuous dynamics are not only existing in humans and not only in fMRI. You can see a lot of these dynamics in different species, different types of modalities like optical imaging, MEG, even EEG back in the day. You can really see those posterior to anterior propagation of waves, even though it was very highly discretized at that point. But that means these things really exist, and we need to find ways to kind of use theoretical framework to describe these dynamics. That's why we have this session. So basically, obviously, as Joanna said, models are not perfect, but some models are useful, and our goal always is to use these models to describe as many things as possible in our empirical data. That's why theoretical and modeling perspectives are, I think they always come hand in hand with empirical observations, especially over the last several decades. And more importantly, what I'll be discussing in this presentation is how does the physical and geometric embedding of the brain can constrain a lot of the things that we see in this data. Right. So we start with neural field theory. And I'm going to do a relatively simple explanation of it, because neural field theory has been there for the last 50, 60 years. And there's a lot of different but common themes of how neural field theory has been developed. But the most common denominator of that is neural field theory is just a class of models, right? It's trying to discuss aggregate dynamics. And why is that useful? In physics, for example, the perspective of mean field dynamics has been powerful and has been used in a lot of various situations. And I always like using this example. if you ask a physicist to model the molecules and particles swirl, kind of in this room, it will be a headache, right? Because there are millions and millions of particles in this room. But there are ways for you to describe the overall dynamics of those particles, which will give you something useful to describe this room, like temperature, right? Temperature is just a result of interactions of those particles colliding and emitting some energy. So if we try to model all those millions of molecules at once, we need high supercomputers. But we can always find ways to simplify the problem and look at the overall dynamics, and we'll still give you a good explanation of your phenomena. And again, same with swarm of birds, right? You can model every single movement of that bird, or of every single bird in this flock, but you can clearly see that overall they kind of follow the same overall mean field dynamics. So you can apply the same thing in the brain, right? You can create mean field quantities that will describe the different aspects of your biological system. So you can look at, instead of looking at the synapses and then the dendritic dynamics of every single neuron, you can look at clusters of these neurons, or classes or populations of these neurons. So that's what neurofield theory is all about. And what I'll be kind of focusing a lot more is the Robinson et al. Neurofield theory, or as John said, the Robinson-Rennie-Wright neurofield theory. Basically, there are 5 different things that this neurofield theory is trying to model. Obviously, the overall activity of a population, A or B, phi. And then we need some kind of biologically realistic relationships between how populations interact and how the activity emerges. One of the things would be, how can you convert the activity to some kind of postsynaptic potential? And again, we're not measuring single neuron postsynaptic potential. What I'm talking about here is kind of the overall, how does the overall synaptodendritic dynamics look like? And there are various ways to model that, and it's not showing in there. But John has mentioned, you can use kind of convolution type operators. And the next thing is, okay, if you have this cluster of populations and it has an overall postsynaptic potential, how can you now model the activity of a soma? Obviously, in this case, again, we're not looking at a single neuron, but the soma of multiple neurons. And you can do that as well in this neurofield theory. And then the next thing is, how do you convert that potential to some kind of firing rate? And John, again, this morning has showed that one of the simplest ways to do that is to create a kind of sigmoidal function that converts potential to some firing rate. And the last bit is our most favorite one, is how do we now convert this firing rate to the activity? And that's where the signal wave propagation comes into play, what Michael Breakspear showed this morning. So basically, we're not only interested in how the populations fluctuate in time, but how does it affect this spatial temporal dynamics of the system. And I'm going to describe more about the wave equation in the next couple of slides. So that's pretty much the basics of this Robinson neurofield theory. But I'll be, can't see. This is the basic kind of proof. That's the core modeling bit of the Robinson et al. Neurofield theory. And now you can decide, okay, how many populations do I want to check? Because obviously, the population A and B are very general. You can describe however population you want. You can describe excitatory populations, or populations in the thalamus, or in the hippocampus, and so on and so forth. So the most basic, the most popular are the Robinson et al. Neurofield theory. Anyway, I think I've Anyway, it's in the next couple of slides. Sorry. One of the also one of the assumptions of the Robinson et al. Neural field theory, that's why we needed that wave propagation, is that it captures a lot of these very localized interactions. So as Victor mentioned a while ago, it captures the intracortical connections. And why is that important? Because if you look at a single layer, if you look at a strip across the layer, you can see a lot of these very local lateralized connections between within a layer, right? And if you discretize the cortical medium or the cortical sheet in chunks, you will miss out all these local lateralized connections. So it is important. They are there for a reason. Evolution has dictated that they should be there. So the question is, what is it doing, and how important are these local and lateralized connections? Another assumption of the Robinson neural field theory is that the connectivity kernel is highly simplified following an exponentially decaying rule. Basically, connections that are close to points that are close to each other will most likely have a stronger connection. And as you move further down, it will get lower and lower, and it follows an exponential footprint. And there's a lot of evidence that this exists in different species and modalities. So one of the easiest thing to do is get diffusion MRI, get the weights between nodes, and you kind of plot the weight between nodes as a function of the distance between those points. And if you do that, and this is in logarithmic scale, you will see this exponential footprint. So basically, most connections will follow that footprint, and there you only have kind of points that will deviate within that footprint. And people have shown that this kind of footprint exists in other types of primates and other animals, say here in the marmoset. So these two assumptions are biologically realistic, and the question is, how useful and important are these two types of connections? So, and then again, the next assumption is that populations will interact in space and time following this damped wave equation. So if you know a harmonic oscillator, basically it will kind of propagate, and then it will get weaker and weaker as soon as it disappears if you don't have an external stimulus. So that's pretty much how populations are related in space in the Robinson neural field theory. And some terminologies. So this is what describes the damping rate, so how fast the decay of that oscillator would be. This is a parameter of how far kind of the relationships between points. And then our most favorite nowadays is this kernel in here, which is the Laplacian, which encodes a lot of those spatial relationships between the points. So this is what I wanted a while ago. This should have been my mistake. So one of the most kind of popular Robinson neural field theory is the cortical thalamic neural field theory. Basically, what does that mean? Using the ingredients that I showed you a while ago, the pentagon, you can create 4 types of populations. So in this neural field theory, you have a population of excitatory population, cortical excitatory population. You also have the inhibitory population. And the Robinson et al. Model has divided the thalamic population into 2. And you can create these lines denote how those populations are connected to each other. And those arrows and those circles denote which one inhibits and excites which population. So each population in here will have all those flavors of average firing rate, average activity, and so on and so forth. And the reason why this has been a popular one, because it has been used to explain a lot of different physiological phenomena, mostly in, back in the day in EEG. So this is one of our works where you can, it fits perfectly. Michael was saying that, that this type of neural field theory has been really, really useful in explaining the emergence of the alpha rhythm. And it fits it perfectly, almost perfectly. And also other types of rhythms that is, again, the emergence of how the model fits the data is not baked in, right? It's just an emergent phenomena of how connections between this cortical thalamic loop is described. And it has also been used to describe how you can stage sleeps into, REM, deep, and so on and so forth using EEG. And the one that I'll be focusing in the next several slides will be how it can describe the eigenmodes of brain activity. I'll describe that later on, and how it can be used to describe standing, traveling, and rotating waves in the brain. Okay, the fun bit. So I'm going to be talking a lot about these eigenmodes, and there's a lot of analogy of eigenmodes in different systems, in both 1D, 2D, and 3-dimensional systems. So the simplest will be a 1-dimensional string, and you've seen a lot of videos of this from Joanna's talk, where if you kind of ping it and let it oscillate, you will see the emergence of these really intricate patterns that stay there, but they fluctuate also in time as standing waves. Again, in this case, in a 2-dimensional system, which is called a Pacladny plate, where you put like a sand in there and then you just vibrate it, you will see the emergence of these patterns, which are the modes of that plate. So the crucial bit in all these systems and the modes of these systems is that it's really sensitive to the physical property of that system. And if there's a musician in the room, you know exactly how, if you play a violin or guitar, if you change the way you place your hand, you will change the notes that will emerge. Now you're basically changing the physical property of that string, right? You're shortening it and you're lengthening the string. So you're basically changing the geometry of that system. And again, these modes are highly related to the standing waves that Joanna was showing a while ago. So how do we derive these modes? There are multiple ways, but in this case, we'll be focusing a lot on how you can derive it from geometry. So put simply, to derive the modes, you just need to solve this Helmholtz equation or Laplacian eigenvalue problem. And this is what we call the Laplace-Beltrami operator. I know it's scary, but in Euclidean space, that's pretty much just the double spatial derivative. And then if you solve, so it's kind of like an eigendecomposition. So you construct, well, I'll show you how to construct this operator in a while. And when you do the eigendecomposition, you will get a family of eigenvalues, lambda, and a family of eigenmodes. So this is what we want as a result. So how do we construct the Laplace-Beltrami operator? Because I'm focusing a lot on the eigenmodes of geometry. I only need structural MRI, so T1 weighted MRI. So you can get that pretty easily using FreeSurfer. So put a T1 weighted image and then run FreeSurfer, and then it will provide you a model of the cortical surface. When I say model, obviously it gives you the typical FreeSurfer model is like 164,000 points. So basically, it will create 164,000 points, and it will describe how those points are connected as a mesh. So basically, points are connected as like triangles to best describe the geometry of that system. You can also do the same for not just the cortical surface, but also volumetrically. So you can get a mask in voxel space, say here a mask of the thalamus, and you know which voxels are within its neighborhood, and how those voxels are oriented will provide you a picture of the geometric kind of embedding of that particular mask. And then what we do next is, this is the theoretical, the general definition of that operator. It's very, very scary. But in principle, because our system also invokes a lot of discretization, because we have to define a finite number of points, you can approximate this operator in terms of the meshes. So you can just kind of relate these points in between. You can find the relation between vertices of that mesh, and you can find the angles and so on and so forth, and it will provide you an approximation of that operator. We have a GitHub page to kind of give all of you these modes pretty easily, but the link is towards the end. Now, what you will get are these sets of, or family of eigenmodes. So in the cortex, you will get these eigenmodes, and the modes are naturally arranged in a very, very particular manner. So this is for the cortex, and that's for the thalamus that I showed a while ago. Obviously, I'm just showing the first 3, and I'm just showing the first 100 in here. And the reason why I said they're arranged in a peculiar manner, they are arranged according to their spatial frequency. What do I mean by that? So the very first few will encode those patterns that have very low spatial frequency. They kind of brain-wide change. And as you move further down, you will get this high spatial frequency or short spatial wavelength modes. Basically, you can now see more kind of intricate patterns that happen locally. And then the important property of this is it's the same with any type of eigen decomposition. The eigenmodes are orthogonal by nature, mathematically. That means, their dot product is 0 between the modes, and they are independent. That means, well, as an example, you're very familiar with the sine and cosine functions. These are orthogonal functions. That means if you take the dot product, you will get a 0. That means then you can't express a sine in terms of a cosine. And the reason why that's useful is because you can now use the modes as what we call as a basis set. And what Johannes was showing a while ago, that means you can use the modes in combination. And you can perfectly reconstruct any data using these modes. So mathematically, that means if you have any data, y, in space x, you can express that as a sum, as a linear combination of these modes weighted by some coefficient a. So that's a signal, that's the eigenmode, and that's some kind of weighting coefficient. And that's, you can choose the number of eigenmodes that you want. And this is very analogous to how you can decompose signals in Fourier space, where you can reconstruct any signal in terms of basis functions, which in this case will be the sine functions. But in our case, it will be the eigenmodes of that geometry. And it's also similar to how, how you can combine colors, right? The red, the green, and the blue are kind of, they're orthogonal functions. You can't express red in terms of green and vice versa. But you can combine these colors to get a new color with just a proper, by choosing the proper weighting. So it's pretty much that same principle. So, but I'm sure if you've seen these modes, they're not, obviously, again, as I said, then these concepts are not entirely new in a general sense. If you've opened a physics textbook, I'm sure you've seen or you've heard what we call as spherical harmonics. So spherical harmonics are pretty much just the eigenmodes of a sphere. And this is the reason why the eigenmodes that I was showing, this is what the spherical harmonics would look like in a sphere. And you can clearly see that they look like the eigenmodes that I was showing a while ago. And this is not a big surprise because the cortex is topologically similar to a sphere. That means if you have your cortical surface, if you blow it, you will get the sphere, right? So they're topologically similar. So that means you can create a mapping between the vertices. This is why multimodal surface matching works for different surfaces, right? We always kind of move back and forth between this mapping between sphere and cortex. Now, the only peculiar thing is, so the spherical harmonics are divided into groups. That means these, so say the first eigengroup in here, those three patterns are kind of, they look similar, right? They're kind of just like rotational representations of each other. And they encode the same spatial frequency. And if you go further down the line, you'll get 3, 5, 7, and so on and so forth. So basically, these harmonics would have the same frequency. So if I say plot the eigenvalue of the spherical of a sphere, you will see that each eigengroup will have the same eigenvalue. Now, the only difference of a cortex is that the cortical folding perturbs the symmetry in your system. Hence, those eigenvalue, as you keep adding folds in your sphere, it will the eigenvalues within each eigengroup will start to disperse. So they start to separate with each other. Hence, you will get eigenvalues that are not the same within each the modes within each eigengroup will now have different eigenvalues. But the theoretical works by Peter Robinson, for example, has shown how you can represent cortical eigenmodes in terms of spheres. If you're interested, I can show you which paper I'm talking about. So again, just a recap of how we can use the modes as a basis set. So again, you have here a spatial map. Now I'm adding a temporal component to it because we also want to represent resting state in both space and time, for example. So you can now decompose that spatial temporal map in terms of the eigenmodes weighted by coefficient. Now, the only difference is if you have a spatial map or a spatial temporal map. So for a completely spatial map, this A will be a constant for every single J. That means you only have this kind of expression of every single mode. Now, if you have a spatial temporal map, that means it's not just varying in space, but it also varies in time. You can extract the same thing, but for every single time point. So basically, your coefficient now deviates as a function of time as well. So you can now have this mode by time matrix. Right, so how do we use, I'll just show you some examples of how we used it. So we've shown in our paper that these geometric eigenmodes can efficiently reconstruct fMRI data quite parsimoniously. So what do I mean by that? So you can use like a low number of eigenmodes, say here 100, to almost reconstruct perfectly the data. And 100 is the reason why I said it's a parsimonious representation. 100 is only a subset of about 30,000 modes available to us. And we've shown that you can do that for every single task, for example, in the HCP dataset. And you can get almost 90% accuracy with just using 200 modes. So again, as I said, you only need 200 out of about 30,000 to reach approximately 90% accuracy. And we've shown that it is statistically better than null models. So obviously, we want to test if the reconstruction is just better because it just happened by chance, or is it better compared to some kind of null statistics. But it's important that we choose the proper null model because we want a null model that preserves a lot of the special autocorrelations that exist in our data, which is very, very true in fMRI in particular. So you can use a variety of null models. You can use, say, the Brain Smash toolbox from Bert and John Murray's group. Or you can use a new eigenstrapping null model, which Nikitas from Michael Breakspear's group will present in this poster and in an oral session on Wednesday. So you can check that. So basically, you create these empirical maps and you kind of create surrogates to compare your reconstructions with. And what we're showing that if you compare the empirical reconstructions with respect to these null models, it is better compared to chance. And you can use the same principle of decomposition of fMRI data to a lot of other diverse maps, say in NeuroVolt. But one of the things that we have found is that if you use this mode-based perspective, what we found is most of the task activation maps that we have in fMRI have a lot of power in the first few modes. That means they're the longest wavelength modes. That means there really is this kind of long-range autocorrelations that happen in fMRI data. Okay, I'll probably focus a lot of this. How long do I have? 5. Okay, so in terms of modeling, instead of just using the modes to reconstruct the data, can we actually use to predict the data? That means we now resort to some kind of modeling framework. So the only thing that we will do is we will use, again, the Robinson et al. Wave model. And that means we're only focusing on cortical-cortical connections. So we've removed the thalamus, the populations in the thalamus. So this is just dynamics within the cortex. So basically, dynamics that is just driven by the geometry of the system. And you can do some mathematical tricks, because obviously, there's a space and time component to it, right? So the problem is relatively difficult to solve. But you can do some mathematical tricks. And if you exploit the idea that the eigenmodes are orthogonal, and in the linear regime, we can approximate activity in terms of separable variables. That means you can separate space and time. That means there's no mixing between space and time. That means you can represent the total spatial temporal activity in terms of a product of time, a temporal variable and the spatial variable, which in this case will be the eigenmodes. And you can do that trick to convert this equation into this equation. That means this is a temporal differential equation, right? That means it only describes the temporal dynamics of every single mode. And that means every single mode will fluctuate in time. And what does that fluctuation look like? It looks like a damped harmonic oscillator. So in the absence in the presence of just an impulse input, for example, this is what the solution would look like. Again, the reason why it's damped is because there's an, okay. So this is just the oscillatory component. That's the cosine in here. And the frequency is related to the eigenvalues of your modes. But the reason why it's damped is because the solution will decay in time exponentially as dictated by the parameter of that model. And then we ask how useful is this model and how can it be used to predict data? And this is just an example of how the wave propagation will happen if you run the model with just a noise as a stimulus. What we found is, I'm going to skip a lot of the detail, but in the modeling world, there are a variety of ways to do comparison between model and empirical data, right? And most of the time, as Johannes said, people use static functional connectivity. So that means you kind of relate edge FC and node FC. But people have now started doing more of comparison, not just static, but dynamically. And there are various ways to do that. One of them is Gus-Deger's FCD calculation. And basically what we found is that the simple wave model performs equally or better than some sophisticated neural mass model with more parameters than ours. That means the waves are giving you something relevant to the data of interest, which is in this case fMRI. And you can use multiple ways as prelude to David's talk, next one, which is on stimulation. You can also do some kind of stimulation protocol, where in this case we just did a stimulation of the visual stimulus, the visual cortex, and you just let it propagate in space and time. And what we found is if you follow the trajectory of the activity along some kind of hierarchy, visual hierarchy, of say the Feldman and Van Essen hierarchy. That means the visual hierarchy represents how the input from your retina will go to your LGN, will go to your V1, so on and so on and so forth. So we ask is, can our model predict that kind of hierarchical activations? And what we found is, yes, we can. And it matches a lot of empirical estimations of that hierarchy using T1, T2 weighted, which again, we're only using a simple model with geometry as an input and nothing else. So as the last slide, and so last two slides, you can apply this in a variety of contexts. Again, you can apply this to different subjects, different cohorts, different species. And you can also run the model and test how wave speeds are different across the different species or across the development, for example. One of the things that you can use the most as well is to describe how you can, so it's like another way to do morphometry, if you are familiar with anatomical morphometry, where you're finding significant differences in between patients and controls, for example. You can do a mode-based morphometry, and we have works led by someone in the lab with that. And so in summary, briefly, I think I've shown some examples on how we can use the modes to kind of describe anatomical constraints on a function. And the good thing about this is you only need T1. It's based on geometry. It's linked to a theoretical framework of neural field theory. And I've shown that you can use the modes and geometry to explain how waves travel in the brain and how it can explain diverse physiological phenomena. And again, the approach is very, you can create different avenues for future research using the same framework. And these are just examples of what we are currently doing in the lab, so make sure to visit, especially these 4 people, which are developing a lot of these techniques in a variety of contexts, from connectomes to development, and so on and so forth. And I will end there. Thank you.